Hi, everybody. Today we're going to talk about in-situ remediation techniques and technologies. So let me share my screen and we will get started. All right. So what does it mean when we say in situ? Well, that means that these technologies are used to treat plumes or contamination in the environment where they reside. So we don't take the contaminant out of its environment to treat it. Instead, we're putting a treatment into the environment so that we can treat it right where it is, and we don't have to remove the contaminant. Now, there's some obvious advantages to this, but there are some disadvantages as well. So we're going to talk a little bit about some of these technologies and the differences that we see um, between them. All right, so here's a good sort of overview of which treatment technologies that are in situ and which ones of these can be used for different types of contaminants. And this might be useful for your project, for example. But one of the things that I want to point out here is they have these little dots here. Each of these dots, if it's a completely filled in dot, then that means that it's not a really good option. If it's a halfway filled in dot, it's an average option. And then if it's a completely empty dot, so a circle, that is indicating that that's the better option for that. And then, of course, there's S's on here. These indicate that it's specific to those types of particular contaminants. So you have to look at which contaminant it is and what chemical type it is to see whether that's going to be effective or not. So if I had to check, and let's say I have a plume that has radionuclides, for example, and we want to develop strategies to do in situ treatment. Well, I'm going to look down, oops, get back to here. I'm going to look down this little column here, and I'm going to look for the one here that is an empty dot. This tells me that it's a better technology, so solidification and stabilization might be an option there. We have an average option, which would be the electrokinetic separation, and then bioremediation depends on the type of radionuclide that we're talking about. And the rest of these are not such good options. So that would sort of limit us when we're discussing what treatment options we have available. Okay, so let's talk about a few of these in depth a little bit. So here is soil vapor extraction. And the way that this works is we often use this, for example, for contaminants that are halogenated or non-halogenated VOCs, that's volatile organic compound, and for fuels. And the things that these have in common is that they're very readily volatile, meaning that they can go into a gas or vapor phase easily from a liquid. And that allows us to take advantage of this soil vapor extraction method where we have injection wells, let's say, on either side here of an extraction well. And then we start to pump in air, let's say, into these outer wells right here, these extraction wells, or excuse me, injection wells. And then at the same time, we start to apply a vacuum at the extraction well where we're trying to remove that contaminant. So what happens is the air comes in, it goes through the pore spaces here towards the extraction well at the center. And as it goes through those pore spaces, it volatilizes these compounds. It picks up the volatilized compound and brings it over to the extraction well where it then can be able to remove it. So this is going to be better suited for things like soil that's well drained because first of all, if you notice this picture here, the water table is below where we have our contaminant at and where we have our wells at. And that's purposefully done because we can't, if this goes into the liquid down here, then we don't have that pore space, that empty pore space where the gases can flow through because that pore space below the water table here is going to be filled with water. So it really only works for above the water table itself, so in the soil area. And it has to be well drained so that there's not a lot of liquid in the pore space. We want it to be highly permeable so that we can have a lot of gas transfer going through the soil. And typically low organic carbon. If we have higher organic carbon, we might have, some issues with more compaction and other things where there won't be that readily flowing air that can go through and pick up that volatile contaminant. You can also do, in some cases, a pulsed pumping strategy. So if, let's say, the soil here, it's diffusion limited, meaning that it takes a really long time or it's difficult to get the air to be transported through those pores towards the extraction well, meaning that it's going very slowly towards the extraction well. Well, maybe it's worth it to just pulse a little bit of air, and then that pulse will go inwards, and then you can pulse another bit of air, and that way you're not continuously putting in as much energy that's not really going to get you a lot more results as far as pressurizing these air injection wells. All right, so another option that we have, this is one that is we saw with the radionuclides, was an option for us. Solidification and stabilization. These are actually two different strategies, but they work in the same manner. So the way that this works is to really hold the contaminant in place. It's not necessarily to remove the contaminant or to destroy the contaminant, but really to prevent that contaminant from moving anywhere so that it can't impact human health or the environment. And so some of the things that we might use this for: inorganics, radionuclides, radioactive, hazardous, mixed wastes, things that can be really tricky maybe to remediate, we can use solidification and stabilization. So solidification, this is where we reduce the mobility of that contaminant by by putting something into the environment that will react with the contaminant matrix, meaning if it's residing in the soil, what we put in is going to be reacting with the soil particles or the sediment itself, not necessarily with the contaminant. And so things like, let's say, a cement where, we are putting that, solidification material into the soil, that is going to react with some of the soil around it. It's going to create like an encased area, and we're going to prevent the mobilization of that contaminant. Um, Similarly, stabilization also prevents movement of the contaminant, but in this case, it's a reaction with the contaminant itself that causes that stabilization. So that might be that we are changing the, let's say, uh, binding capability of that contaminant. So for example, we might have a chelator. So chelators, Chelator, that word means something that's going to reach out and grab and bind onto metals in particular. And so we have a lot of chelators and different things, like even our shampoo, one of the most common being EDTA. And this type of chelator is really designed to grab onto the metals, like magnesium and calcium that are in hard water, and allow you to be able to get, your hair washed a little bit better, even if you have hard water in your home. So that's what those are for. So you may see EDTA on that sometimes in other, hygiene products and things like that, that is designed to really counteract the effects of hard water on the reduced kind of bubbling and other things that you want to see from your soap and from your shampoo. But in general, chelators can also grab on and bind different contaminants. They might be able to bind onto those contaminants and also bind something else. And they might just make it so that, those contaminants are no longer reactive with other things because they're so tightly bound to that chelator. So that would be an example of stabilization as opposed to solidification, which would be more like pouring in, let's say, the Portland cement or the binders or something like that, that's going to react with the soil matrix around it. Now, we do have to keep in mind that a lot of organics will interfere with the hydration process and impede the curing of cement. So this can be a challenge, especially if you have a lot of organics in your soil or in your sediments. And so that has to be considered at the site where you're trying to do either solidification or stabilization. So in this, one of the biggest challenges is really trying to get a good mix of the binder and the contaminant in the matrix, right? And by matrix, we usually mean soils or sediments or something like that, right? So there's a few different, mixing approaches, but the most common being something along this lines using an auger. This drills in and it has this auger that will release binding agents into the soil, and then the augers will spin and mix it until that's all hopefully very well mixed, and then they can pull that out and the soil will solidify or the contaminant will solidify in place. All right, so next up, we're going to talk about chemical oxidation. So this is redox reactions. So it can be reduction or oxidation, but very commonly oxidation that is used to convert hazardous contaminants into less hazardous or non-hazardous, or types of contaminants that are less stable, less mobile, or rather more stable, less mobile, or inert. So we want to basically either remove the harm of the contaminant itself or turn it into something that's going to be less harmful or less mobile and therefore less likely to impact humans or the environment. So this is one that you're going to be actually, calculating for your project. So also things like Fenton's catalyzed hydrogen peroxide, hydrogen peroxide on its own. Ozone is another one. This is a gaseous oxidant, so we can use that a little bit differently. And then sodium persulfate. So these are all pretty common oxidants. And the idea is that we are using them to oxidize the contaminant, making it in a, less harmful state. And so for these, if we're treating it in situ, what we're doing is we're putting in all of the, um, let's say we're using something like sodium permanganate. What we're doing is we usually have it in a liquid mixture. We're putting it into the into the ground or into the subsurface, and we might be mixing that around with some augers, some backhoes, other things like that. If we have liquid or in some cases solid oxidants that we're using. We can also use injection for liquid oxidants. So we could use wells or trenches there. We can use pressurized injection of liquid or gaseous oxidants. And then in some cases, some hydraulic fracturing techniques for pushing solid oxidants into place where they're needed. This varies a little bit differently from your project because in your project, I am asking you to pull the contaminant out and then use the chemical oxidant to treat the contamination. So we're actually kind of considering it as an ex situ option, but there is an in situ option as well, which is this chemical oxidation in situ, meaning that we can put it in place, that oxidant in place, and oxidize it where it's at. Now, there are some concerns around this method of doing oxidation. So first of all, we have to ask whether that oxidant is appropriate and whether it needs a catalyst. So will this oxidant that we picked actually be effective in oxidizing the contaminant? Second, when we're doing this in situ, and this is not a concern if we're doing chemical oxidation ex situ, but we want to know what is the soil oxidant demand. So as you can imagine, if we just put this chemical oxidant into the soil, there's a lot of other stuff there, including organics and humic acids and other things that this oxidant is going to react with. And if we want to make sure that we're going to have enough oxidant there to get rid of our contaminant, we need to make sure that we are first satisfying the soil oxidant demand, which basically means that we are putting in enough oxidant to fully oxidize everything else that's in that soil. And then on top of that, putting in enough oxidant to be able to oxidize the contaminant that we have that we need to get rid of. So here, this is going to make our calculations of how much chemical we actually need be much, much larger than if we do this chemical oxidation ex situ. Because if we do it ex situ, that means that we're first removing that chemical or that contaminant, and then we're treating it outside of the environment. When we do that, we're really only extracting the soluble, liquid fraction, right? So we're not bringing with it all of the soil particles that are going to be exerting this soil oxidant demand. So in that case, we may consider, if we're going to do chemical oxidation, we may consider whether it's less expensive or easier to do ex situ and have fewer chemical costs, but now we have to do like pumping and having a different tank to treat this in and so on, or whether it's easier to just put more chemical in, satisfy the soil oxidant demand, and then also satisfy the demand for the oxidant from the chemical that we're actually treating. So we have to consider this SOD if we're doing it in situ only. All right, next up, we have to also consider pH because pH makes a big difference in how effective our oxidants will be. Some oxidants like a more acidic environment, some more basic, and so we have to make sure that whatever we do select is going to be appropriate for that environment and that pH because we're not very easily able to adjust the pH of soil or groundwater. And then also, we want to think about the decomposition rate because in some cases, if it's a very slow process of oxidizing, then maybe these oxidants might remain there for a long time. Others, like ozone, are very quick. They go in, they decompose, and then they're gone. But if we put in a chemical that's going to be remaining for quite some time, how will that affect our soils, our environment, other things around there? Or even if we have a different second remediation option that we want to use, like bioremediation, would this be something that would impede that if we put this into the environment? So we have to think about that as well. All right, next up is a zero valent iron permeable reactive barrier. And you are going to be looking at this as well for your project. So this is how the zero valent iron reactive barriers work. So what we do is we first see that there's this plume right here, and we see it has a bunch of things in it that can be uh, removed, or let's call it decomposed, by using a reduction technique, meaning that they are going to be, if they're going to be accepting electrons, and that will help them either decompose into something else that's less toxic or turn into something that's not as mobile in the environment. And so what we do is we see, okay, this groundwater is flowing in this direction here. Let's get ahead of the where this plume is moving to. And so we're going to make a trench, and we're going to fill it with this zero valent iron that's kind of like in a powder form, so it has a lot of surface area on it. So we fill this trench up with this ZVI. You can see down below here, this is an area where they're making one of those trenches and filling it up. And then as the plume goes and travels, it starts to travel through this permeable barrier. And as it goes through this permeable barrier, a couple of things happen. First of all, the microbes that live here could be potentially using the zero valent iron as a source of electrons to be able to do microbial decomposition of these components. They can also be decomposed abiotically in some cases using electrons generated from this zero valent iron. And thirdly, we might just be changing for example, in this chromium case, we might be changing the nature of the chemical so that it's in a less soluble form. And so how does our ZVI do this? Well, it is a really interesting type of uh, reaction that happens. I'm going to try to draw it for you. So in the subsurface, or in sort of oxygen deprived reducing environments, zero valent iron, which is really just Fe0, right? It's metallic iron. This gets changed, and basically it undergoes what we call anaerobic corrosion, meaning that it's a non-oxygen needing corrosion process. And this happens where our Fe0 turns into Fe2 plus and 2 electrons. So this is our half reaction for our zero valent iron oxidation half reaction. In cases where there's liquid present, this can then be related to a water reaction where now the water electrolysis is happening, you're getting hydrogen out. So instead of these 2 electrons here, we may be getting hydrogen that's carrying those 2 electrons. And so either way, a lot of different microbes and even some chemicals can be reduced or given electrons from either just straight electrons or this hydrogen that's produced in the water itself. So this is something that can happen and then allows us to look at the option of using just kind of this passive barrier here to treat the plume as it goes through so that we have this treated groundwater coming out the other side, and then no more plume or other contaminants are there, or at least they're very well reduced. Oops, let me erase this. It's not on the next screen here. All right. Okay, so next up, we're going to talk about soil flushing. This is exactly what it sounds like, essentially putting a whole bunch of a liquid of some kind into the soil where the contaminant is at. And then we're going to kind of wash it through the soil. The contaminant will get into the liquid, and then all of that will be brought using a well generally up to the surface for disposal or recirculation or treatment or other things. So what this tries to do is do solubilization of the contaminant itself, help form kind of these emulsions. So even if it's something that, let's say, is not super soluble, we might be able to put in a type of chemical, a type of liquid, let's say, that can help it become more solubilized and will grab that contaminant and then bring it in the liquid phase out. And we can also even pick some chemicals that, or some flushing solutions that will have chemical reactions with our contaminant to help reduce how much of it is left over at the end as well. So the different flushing solutions that we typically will use, water. This is great for anything that's hydrophilic, so water-soluble contaminants. Acids, we might use this for metals or basic organic materials because these can be solubilized, just like when we talked about the landfill. We had a certain period where when the pH dropped, that we started to see metals come out in the leachate, and that was because the metals were becoming solubilized and being carried with the liquid to the bottom of the landfill. But then again, once we increased our pH, then we didn't see those metals anymore because they reprecipitated back out and became trapped wherever they were at in the landfill. In some cases, though, however, bases can actually act as solubilizers. So things like zinc, tin, or lead, and some phenols tend to be a little bit more soluble in basic pH. Other chelators, complexing agents, reducing agents, these things can be good for metals. And co-solvents might be needed for some organic materials. And then surfactants. So if we have something that's hydrophobic, rather than putting in water, which they're not, that's not going to pick up the hydrophobic contaminant, we might have to put in a surfactant, which acts sort of like if you will, like a soap, and brings that contaminant into that solution and allows it to travel and become flushed or kind of rinsed out of the soil with this soil flushing technique. Okay, so electrokinetic separation. This is a really neat one. This is where we put electrodes into the ground, attach them to a power source, and one electrode is positive, one electrode is negative, and then we apply a low intensity direct current through the soil, and that polarizes. So we have, polar electrodes, which can help mobilize ions, for example, to go in one direction towards the opposite charge. So, opposites attract. So if we have a positively charged electrode here, our anions are going to be traveling towards it. And then if we have a negatively charged electrode over here, we're going to have our cations or positively charged ions traveling towards that. So this is one thing that is really an interesting separation mechanism because it doesn't require advective flow, meaning it doesn't require the water in the pore space to flow from one area to another. In fact, it's just attracting the ions and it's not moving the pore water at all. And so this can, be good. This can also create a few challenges. So if we have really kind of fine-grained soil, in some cases, we can actually achieve electroosmosis, which is sort of this electrically induced hydraulic flow of the ground or soil pore water in between the electrodes. And so this is where we start to see the soil almost acting as like a membrane separating the anode and the cathode. And because of this, we have, essentially generated an electrically induced flow of water that can go and even carry some neutrally charged species with it as well. This is in more of the fine-grained soil as opposed to kind of the bigger pore space soil. So then we can also get charged colloids in mice cells moving as well. These are kind of like we can think of these as small particles, like colloids, generally negatively charged, and then micelles, which can have different charges on the inside versus outside. And these can move through this process of electrophoresis, so going towards the certain opposite charged electrode, but it's now kind of a coagulation or a bunch of particles or a bunch of things rather than just a single molecule or single ion. It may have a few different species attached to it. So with my cells are interesting. They have generally this kind of, um, they can have one hydrophobic end and one hydrophilic end, and they can go together in chains like this, but that also allows some kind of a thing that will be trapped in the middle, let's say. And let's say we have a positively charged um, on one end and a negative charge on another. Well, oops, negative charge here, negative, and then here we have our positive charge. Well, then we could have something that's negatively charged here attached through all of these right here, and then we can have that travel as if it's one big charged piece. So these electrolysis reactions, they do create hydrogen and OH minus at the cathode, and they create oxygen and protons at the anode. So we have to be a little bit cognizant of that. We don't want to be creating a whole bunch of oxygen and hydrogen and then have some sort of a spark that creates an explosion. So we have to be very careful of that. They also can create acids near the anode and then base near the cathode, and then those are going to kind of migrate towards each other. And the acid front will increase the mobility of the cationic species, but in some soils it can prevent this electroosmosis from happening very well. The hydroxide front, the OH minus, that needs to be controlled so that we don't end up having the controlled precipitation of some of the target metals that we want to be maintained in the acid front and allow them to be solubilized in the acid. If we have the hydroxide front move too quickly, it can catch up to that and sort of precipitate out our metals rather than allowing them to continue on to our electrodes. Okay, we can also do thermal processes. So the addition of the heat to the soil can help us remove some of the volatile or semi-volatile contaminants. So we can do things like electrical resistance heating, steam injection, conductive heating, or even using radio frequencies to heat this. This allows for better extraction through vapor extraction and or liquid extraction in some cases. So here, what the idea is to just heat it up so that we can increase the volatile components of the contaminants, meaning that we're driving it into gas or vapor phase, and then we're able to collect it a little bit better. Okay, and then my favorite is going to be bioremediation, and this encompasses a large number of different mechanisms of doing bioremediation. So I'm going to give you just a few of these pieces. So we have a few different terms associated with bioremediation. If what we're doing is adding microorganisms that are missing, let's say we have some unusual pipeline of a chemical that's running from state 1 to state 2, and it breaks halfway between, and it's now going all over the soil. But because that chemical doesn't generally exist in nature in that area, maybe the microorganisms that can break it down are not there. Maybe everything else is. And so what we want to do is do bioaugmentation, where we put enough of those microbes, the right types of microbes there, to be able to break down the contaminant. This is not as straightforward as you might think, however, because a lot of the microbes that we want to break down our contaminants, they might have a very niche environment that they can live in. And they are not used to, if they're thrown into a whole brand new environment, they're not used to competing in that environment with the native microbes that are there. So in a lot of cases, if that microbe is slow growing, or if it's not able to keep up and compete or outcompete the other kind of native microbes that are already in place, then sometimes bioaugmentation doesn't work. So it's not always just about finding the right microbe, because also creating the environment is just as important so that they can do their job in removing that contaminant. So in a lot of cases, what we do is not so much bioaugmentation as we do adding things that are missing that will allow native microbes to do what they can already do and remove that contaminant. So here, for example, aerobic bioventing, we might pump oxygen into the subsurface if this is something, let's say, that can only be degraded aerobically, and we know that the microbes are there that can do it. They just don't have that electron acceptor. So we can add that electron acceptor for them and allow them to do what they naturally can do. We can, in some cases, do anaerobic bioventing. This is not as common, and I'll say the reason for that is, first of all, a lot of the subsurface soil where we do a lot of this bioremediation, a lot of that is mostly anoxic or anaerobic already. So in that case, microbes are already doing anaerobic processes or anoxic processes, and they might already have everything they need. They just might be operating a little bit slowly. Now, things that we might put into the subsurface if needed, nitrogen, in some cases, very few, that can be needed for some nitrogen needs or for certain root nodule microbes that do nitrogen fixation. So that could be something that we might put there. Hydrogen, although this is a really great electron donor and might help in soils where they have plenty of electron acceptor, just not enough electron donor, that can be super helpful. However, the explosion risk and the fire risk is really concerning, especially when you're pumping it into the subsurface and you're not sure exactly whether that might travel into somebody's basement or other, crawl space of their house, something like that. You don't want that to happen. So the hydrogen tends to be used very infrequently, and more so if this is needed, they would rather put zero valent iron, where this is, produced at a less less quick pace, or let's say, and just supplies it as needed rather than putting in the actual hydrogen itself. And then carbon dioxide, this could be something that might be needed in some cases, especially if it's autotrophic growth that requires carbon dioxide for cell building. But again, that's not as frequent as something like aerobic bioventing, which just speeds up a lot of the aerobic processes for things there. We can also do co-metabolic bioventing. This is where we're putting in something that's not necessarily an electron donor or electron acceptor, but something else entirely that might stimulate a response from the microbes to generate or to produce enzymes that are also used to break down the contaminant. So it might be that we are stimulating the production of a certain enzyme by putting certain things into the soil or subsurface, and that enzyme, even though its main purpose is to break down what we're putting in, it also might have a secondary purpose and be able to break down our contaminant as well. And so even though we're not putting in something that's specifically being used to break down the contaminant, it is stimulating the production of enzymes that can be used to break down something else completely, this contaminant that we want to remove. All right, so one kind of thing that we do see in especially these kind of bioremediation processes, which is also related to a lot of what happens with zero valent iron impermeable reactive barriers, but this is a microbial mediated breakdown of PCE and TCE, the dry cleaning chemicals that you've probably heard me talk about a few times in class. So PCE looks like this. It's two carbon compounds or two carbon molecules that are double bound together, and they each have two chlorides associated with them, so 4 total per PCE molecule. And you can see here that it does take electrons and protons to be able to strip off 1 chloride. Each of these steps strips 1 off. And what one of the problems is with this reductive dechlorination pathway is that we might get stuck somewhere here because each of these steps requires electron donor. And if we don't have a lot of organic material, we don't have a lot of, let's say, decaying matter or other means of electrons in our soil, then we might only get 1 or 2 steps, and we might not get all the way down to ethene here. The problem is that if we stop here at DCE, dichloroethene, or if we get to vinyl chloride right here, either of these can be more toxic than the original parent compounds, PCE and TCE. So we've actually made things worse if we don't make it completely through the pathway of reductive dechlorination all the way to ethene here. So we need to be sure that we're adding in enough electrons to get through each of these steps so that we can get completely to ethene, which is non-toxic and will be fine for the environment. So in general, what we can do is look at bioremediation in terms of how far away from the site or the contaminated zone or area we are, and what might actually be predominating in each of these zones. So we'll see that we have a range of different metabolisms that are involved based on how far away from the original spill site there is. And of course, we have the spill site right here, and we have groundwater flow going in this direction. Well, if we look way far out where we're not even near the plume, well, then we might have plenty of oxygen present in the groundwater due to just diffusion and other things like that. So this could be an area out here where aerobic respiration could take place using oxygen as the electron acceptor. But once we run out of the oxygen, so let's say we move a little bit closer, and now we're in this zone of the plume, so we're in this kind of outer ring here. And here, the microbes have already consumed all of the oxygen, so now they're on to their next best electron acceptor here, this nitrate. And so this is an area where nitrate reduction is happening. So different microbes will be doing nitrate reduction here as just like different microbes will be out here doing aerobic respiration. Then as the plume keeps moving, we enter into this manganese reduction area. So we might have manganese being reduced. And then when we run out of manganese, we might have iron reduction occurring. And when we've run out of that, we'll have sulfate reduction occurring. And then when we run out of all of our exogenous electron acceptors, we're going to start having fermentation and methanogenesis happening, which is what we would expect to see close to our spill site, because that's where we have the highest concentration of our contaminant. And it's also where likely any oxygen or nitrate or other electron acceptors has already been consumed because the microbes there will have used those things up first and trying to consume, that very concentrated contaminant will create that kind of anaerobic, if you will, reaction zone right up near the spill site itself. So this is a good way to look at how the different microbes are all probably involved in different pieces of bioremediation at different locations along the plume based on, where they are, what electron acceptors are present, and how this groundwater in this plume is moving. Okay, one more type of bioremediation, or actually two more types. This first one is phytoremediation, and this is where we use plants instead of microorganisms to do remediation of different contaminants. And so we can do various different things that can happen within phytoremediation. So we can have phytodegradation. This is where the plant itself is breaking down that contaminant. We can have rhizodegradation. This is where the root zone and the microbes in particular around the root zone can do degradation of that contaminant. We can have phytoextraction, which is just basically pulling that contaminant up into the plant material. It doesn't break it down, but it does kind of sequester it in the plant material. And then if you want to remove that contaminant from the environment, you have to simply remove the plant material. And then the plants have helped you extract that from the environment. You can have phytovolatilization. This is where now, once that chemical has been taken into the plant tissue, it can be volatilized off of that tissue in sort of the same form that we have transpiration that happens off of, vegetation. And then we can also have phytostabilization, which is kind of the same thing that stabilization is to us earlier in the presentation. But in this case, it's just that something associated with the roots or the root nodules or the plant itself is causing the reaction to stabilize that contaminant, maybe changing its redox properties, maybe changing its characteristics, but somehow stabilizing it so it's no longer moving through the environment and no longer going to be a danger. Another kind of final type of bioremediation is what I call microremediation here. This is fungal bioremediation. And down here at the bottom, you can see this is the turkey tail fungus, or Trametes versicolor. You can grow this from what is called mycelium. This is this little fuzzy looking stuff right here. And it grows through this tree and these logs and so on, and then creates these little fruiting bodies, which are the mushrooms that we see coming out of the tree log. Well, this is a really cool type of fungus because it's type of white rot fungus. And that means that when you go into the forest, if you've ever seen a log that's rotting away and it looks all white and kind of, like it's kind of like, spongy and white. That's because white rot fungus has been able to consume all of the brown lignin in that log, in that tissue, that plant tissue, and leave behind the white cellulose, which is what gives it the name white rot fungus. So it means that, what we're seeing is the white cellulose left behind. But before they knew that, they just referred to it as white rot fungus because that was what the coloration looked like when it was colonizing a log. Well, these types of white rot fungus, like the turkey tail here, they are really special. They have a lot of different cool enzymes, including lacase, manganese peroxidase, and lignin peroxidase. And all three of these enzymes are extracellular enzymes, meaning that the the fuzzy mycelium here excretes these enzymes outside of their cells and can break down a wide number of contaminants that are really impressive. So you can, for example, for a research project when I was an undergraduate student, I looked at how this type of fungus could be used to decolorize two different types of synthetic textile dye. And it can do that through these sort of non-specific enzymes that it excretes and can break down a wide number of things. In fact, the lacase from this type of fungus, this Trametes versicolor, the lacase enzyme is used in pulp and paper pulp and paper industry to be able to do enzymatic bleaching of the pulp because it allows for the enzymes to eat that brown lignin and leave behind the white cellulose and thereby do sort of enzymatic or bio bleaching of the pulp without having to use really strong and harsh chemicals and other oxidants, right? And so this is one thing that's used currently and also will be, explored for a lot of other reasons, including remediation of contaminants, where you can see here on the right, they've put colonized logs. So these logs have been pre-colonized with this mycelium, and they can put them into the ground so that they act as sort of a filtration or barrier. And as contaminated liquid goes through that soil, then the mycelium and the enzymes associated with it can break down those contaminants. So those are, it's a really cool area of research, but also one that's been used successfully to do some bioremediation as well. All right, and then finally, we have what is called monitored natural attenuation, and this is what we call the watch and wait approach. And although it might seem counterintuitive to think of this as an approach to actually, removing or remediating a chemical, it is a very valid approach if there are certain things that are certain criteria that are met. So number one, we want to make sure that it's not an immediate risk for human health or the environment. Number two, we might be just watching and waiting because let's say the microbes are there, the necessary nutrients are there, and maybe it's just a matter of time before the native microbes actually take care of it, or through some other process like UV or some other method or mechanism that contaminant gets removed. But third, it might be that one of those methods that we've already applied it just takes a long time, especially in bioremediation. It's not an instant fix. And so we might think that, okay, maybe the best option is to do nothing. Some cases, this can also be the case if it can lead to bigger harms digging up a site, let's say, that has a bunch of really hazardous material that might then, get out of the area that it's currently in and maybe expose people, workers, and other, maybe sensitive environments and so on. So in some cases, we want to just let it be and not disturb it and watch and wait and monitor it and hopefully, if it's working, then it will eventually go away through usually bioremediation or another kind of degradation mechanism that's already in place. And we just have to wait for that to occur. But sometimes this is the right answer. And this is something that should always be considered when we're considering all of the different remediation methods that are out there for a certain site. We should always consider it as an option and see it may be a bad option, maybe a good option, might be the best option. So we have to keep an open mind. And even though it's a little counterintuitive to not just go in and clean something up, sometimes this can be the best option out there. All right, that's it. So thanks for listening, everybody. I hope you all have a great holiday, great Thanksgiving. I will see you back in the classroom on Monday. Have a great week, everyone.